Now we will going to discuss about chromosome walking. So before going to discuss about chromosome walking, I would like to recommend you to go and watch the genomic library video if you do not know what is genomic library and how it is constructed, and I would also like to recommend you to watch the colony hybridization and plaque hybridization and colony PCR videos. Okay, if concept of those particular topics is clear to you, then learning chromosome walking and understanding chromosome walking will be easy. Okay, so let's now consult, or let's try to suppose a hypothetical situation where suppose there is a particular genetic disorder in human. Okay, and we all know that genetic disorder means it is due to because of some gene. Okay, but we don't know anything about that particular gene. Okay, and we also don't know about where in the entire genome that particular gene, let's say gene X, is located. Okay, then how can we study about that gene, and how can we determine how can we determine some solution about this, right? That means I want to told you that there are various kind of genetic function which is coming from what gene is not cleared. Okay, so under such circumstances what you have to do, because remember, you don't know about anything of the gene, and you also don't know where your gene is located in the entire genome. You have the only knowledge that yes, this is coming from that particular organism. Okay, now what will be, so what to do now? Okay, so the idea will be, as we know about what is the organism, right? So our first goal will be to determine the initial or the general location of the gene on the chromosome, because we have the entire genome, right? So suppose we want to find out a gene which is responsible for a genetic disorder in human. Okay, so for you as a human is a eukaryote, so there is a cDNA library for human. Okay, cDNA library is what we will going to discuss about this in our upcoming videos, but for now just understand that cDNA library is exactly the gene that what we have discussed about genomic DNA library. Okay, that means here also we have a petri plate, and in the petri plate we have millions of colonies, and each of the colony will contain clone of cells derived from a single ancestor cell and contains multiple copies of a particular stretch of the DNA. Okay, that means all the colonies together will construct the entire genome. Okay, now you have to find out what is the location of your gene of interest and also what is the sequence. Okay, so I hope you may think about normal screening, right? That means colony hybridization or plaque hybridization, because but remember, I will again going to give you the example that suppose you have to you are requiring some books from library, but you don't know what you require. Okay, that means what is your desire you don't know, then how can your librarian will help you? That means you at least have some basic knowledge about what is the name of your book, or the book is written by whom, or what is the accession number, what is the publisher of the book, right? So that your librarian can help you to find out the book. Similarly, colony hybridization and plaque hybridization is only possible whenever you know at least something about the sequence of the gene of interest. If you don't know about the sequence of the gene of interest, then colony hybridization or plaque hybridization in that case cannot help you. Okay, so the way of colony hybridization I am referring as CH, and plaque hybridization is not possible. Okay, so we have to think about something new to identify the gene of interest and to sequence them. So let's see how we can do this. So first of all, suppose this is the entire genome. Okay, so we can at least have the ability to determine what is the general location of the gene. Okay, that means where my gene of interest can be situated inside the genome. Okay, suppose this particular portion is the location where I can expect my gene of interest can be. Okay, then how can we determine those kind of particular locations? So that can be done with the help of various kind of pedigrees, various kind of crosses that will help us to find out the recombination frequencies. Okay, and the concept of recombination frequencies will help you to construct a map. Genetic map means a idea that will help you to determine which genes is after what. Okay, so you can construct a genetic map. I am again repeating the entire procedure. The idea is we have to first find out what is the particular generalized location of our gene of interest. Okay, and that can be done with the help of some crosses or pedigrees. That cross or pedigrees will help us to find out the recombination frequencies, and these recombination frequencies will help us to construct a map. That map will telling us the entire type of genomes that are present, genes that are present in the genomes. But remember, the map will not going to tell us about which genes, they will not going to tell us about the sequence of the genes. That means let's consider the example of E. coli. Okay, so if we map the entire genome of E. coli, we will get something like that. This is the gene for Z, that is lac Z. After that, this is the sequence for lac Y, and this is the sequence for lac A. But we will, and it is totally depending on the type of the recombination frequencies we have obtained. Okay, but the genetic map will not going to tell us about the sequence of the Z, the sequence of the Y, the sequence of the A. Okay, that means it will simply tells you that which genes is located after what and which genes is located then. Okay, but they will not give you the details of the sequence. Okay, so by constructing the chromosome map, we can find out the neighboring genes that are present in the entire genome. Okay, that means let's come here in this slide. Okay, so suppose this is the entire genome of a human. Okay, and this red marked portion is my gene of interest. I want to find out this one and its entire sequence. Okay, now with the help of chromosome map, I have find out that the gene A is the nearest sequence which has been previously sequenced. Okay, and that is the another important criteria for performing chromosome walking. If you are dealing with a totally new genome, then chromosome walking also cannot help you. For doing chromosome mapping, it is necessary to there will be some already sequenced gene present in the nearby. Okay, so here the gene A is the nearby sequence that is previously cloned and sequenced. Okay, and I want to told you another important thing that I have repeatedly told you during the genomic DNA library lecture, that we have to construct genomic DNA library in such a way that each of the cell will have overlapping fragments. Why? Because now those overlapping fragments will going to help you. Let's see how. So the idea is, after determining the neighboring gene from the gene of interest, this is my gene of interest, we will first look the sequence of the gene A, or you can say clone A. Okay, now on the basis of the its 3' end, we will construct a complementary single stranded DNA sequence, and that will be attached with a p32 at its 5' end. Okay, now as I have told you that genomic DNA library will contain overlapping fragments of DNA, that means what? That means simply that this 3' end of the gene A should have some sequence that is far off the 5' end of the gene B, because that is the actual meaning of overlapping, right? Let's see this picture. So that was the picture I have used during the genomic library explanation. So suppose this is the gene A that is previously cloned, okay, and this is our gene B, and that is the significance of overlapping, that if we construct a complementary single stranded DNA on the basis of its 3' end, can it find the clone B, right? Because we have generated a complementary probe that we are applying again with the help of colony PCR or any kind of stuff, then it have the ability to find out the 5' end of the clone B. Because again, try to understand that I have constructed a single stranded DNA which is complementary to the 3' end of the clone A, okay, and we have constructed the genomic DNA library in such a way that it should have overlapping fragments. That means what? The 3' end of the clone A should be equal to the some stretch of nucleotide which is at the 5' end of the clone B, right? So if we are making a complementary sequence from the 3' end of the clone A, this complementary DNA sequence or the probe can help us to find out the gene B, isn't it? So that is the idea. Now let's come back here. So that is the idea. This probe will help us to find out the gene B, okay, then we will isolate them, then purify them, then we will sequence them. Then if we are getting our gene of interest, then our entire experiment will be stopped here, will be completed here. But if we are not getting our gene of interest, then we have to again make a complementary single stranded DNA that is specifically complementary of the 3' end of the B, and that is as well as also complementary for the 5' end of the C, and it will help us to find out the gene C. And from that gene C, we will again make a complementary after isolation and purification, and then it will help us to find out our desired gene of interest. Okay, that is how, by using chromosomal walking technique with the help of already cloned and sequenced gene of interest, already cloned and sequenced neighboring gene of the gene of interest, we can find out our gene by systematically walking through the chromosome. So that is the overall idea about chromosomal walking. Okay, so with the help of this method, various kind of human genes and other organism gene has been isolated and sequenced. Okay, so again I am repeating the entire thing, that suppose you are finding out a gene, okay, of which you don't know about anything, okay, you just know about its function. Okay, that means suppose a particular genetic disorder is going on and you are wanting to find out which gene is responsible for that. Okay, So first thing is that from that particular organism genomic library, you have to construct a genetic map. Okay, that can be done by using crosses and pedigrees that will help you to find out the recombination frequencies. Okay, these recombination frequencies will give you the maps. And again, I am recommending you that recombination frequency will not going to give the sequence. Okay, mind it very carefully. After that mapping, you will get a overall idea about the generalized location of your gene of interest. Okay, and by looking at the map, you can find out which neighboring gene was previously sequenced and cloned. Okay, now you will use that neighboring gene to find out your gene of interest with the help of a method called chromosomal walking. How it is done? Let's see. So here the gene that was previously cloned and sequenced is gene A. So we will make a complementary single stranded DNA which will again near about 15 to 20 nucleotide long and complementary to the 3' end. We will construct and we will use this probe to find out the B, because I have told you that genomic DNA library is based on the construction of the entire genome just in the form of colonies where all the cell will contain overlapping DNA, right? That means 3' end of the A gene should have some sequence which is complementary, which is the 5' end of the gene B. Okay, so now with the help of that complementary sequence we can find out the gene B. Then we will isolate them and purify. Then on the basis of the 3' end of the gene B, we will again construct a complementary single stranded DNA. And now on the basis of that complementary probe we can find out the C, and from that C again we can find out our gene of interest. So that is how chromosome walking will be done. Remember, one clone at a time. Okay, so that is how chromosome walking technique is going on. So I hope this video will be helpful to you to understand about the basic concept of chromosomal walking. So thank you for listening to this class, and if you find it helpful, so please like the video, share with your friends and subscribe to my channel to get more videos like that.